People still joining. Hold on one second, Dr. Bolshani.
Absolutely. I can see the numbers increasing. So
good turnout today. There we
that's
go.
Good sign.
Yeah, it is. Give it a few more seconds. Okay. Okay, I think we'll go ahead and get started. Please allow me to introduce myself. I'm Dr. Rob Burgess, and I head up business development for Sino-Biological. And it is my pleasure to welcome everyone today to the next installment in the Sino-Biological hosted webinar series titled Lock and Key Immunodetection. I also would like to welcome our designated speaker, Dr. Ashkan Gulshani, who is a professor and the director of the Systems Biology Group at Carleton University and co-founder of Designed Biologics Incorporated. We really only have one main housekeeping item today, and that is I would like to ask all the participants to think of some great questions for Dr. Gulshani, and please ask those questions in the chat box. We'll hold off on answering any of them until the end of Dr. Gulshani's presentation, and then I will run through the questions in the chat box for him and try to get as many of those as possible answered in the time allotted. So with that, it's my pleasure and honor to introduce to you today formally Dr. Ashkan Gulshani. Professor Gulshani obtained his bachelor's from York University and his doctorate from the University of Toronto in Canada. After spending 2 years in Banting and Best Institute of Medical Research, he moved on to Carleton University in Ottawa, where he is currently a tenured full professor and the director of the Systems Biology Group there. He is also a co-founder of Designed Biologics Incorporated, which is a startup company that focuses on the development of alternative peptide drugs, which we're going to hear a lot about today. Really, for the past 20 years, Dr. Gulshani has been investigating the biochemical and biophysical properties of protein-protein interactions and their alternative modes of binding. He currently oversees a full functional genomics and proteomics laboratory. He has authored more than 100 research publications and led a team that computationally predicted the first protein interaction map of a cell. He's been involved also in the characterization and naming of more than a dozen previously uncharacterized genes. And the title of Dr. Gulshani's talk today is Utilizing Non-Conventional Protein Interactions for the Design of Peptide Drugs. So Dr. Gulshani, it's a pleasure to have you today. And I will now turn the screen over to you.
Thank you. It's a pleasure to be here. So just give me 1 second while I'm sharing my screen. Perfect. So again, thank you very much for inviting me to be here to present some of the data that we have gathered over the past number of years. And thank you very much for your very kind introduction. So due to the diversity of research interests that exist in seminars such as this, I've tried to put the talk together in a way that would be of interest to the majority of the audience. By no means I'm trying to insult anyone's intelligence. Simplicity is just for clarification purposes. Now, having said that, I have divided the talk into 2 sections. In the first 20 minutes, I'll be talking about non-conventional protein-protein interaction, which is more of a discovery-based research, more pure research. And in the next 20 minutes, I'll be discussing the application of such knowledge. In my opinion, these 2 sections, they always go hand in hand for 2 reasons. One of them is that applied research, generally speaking, it's driven from discovery-based research. And reason number 2 is that one of the main reasons for the failure of a number of clinical trials, if you ask me and a number of other scientists, is due to the lack of clear understanding of the biology of the disease, as well as the mode of activity of the compound. That leads into flaws in the design of the clinical trials. Okay, so having said that, I'm going to start with some very simple and basic definitions and slides. So why do we study protein-protein interaction? I'm a protein-protein interaction guy. So why do I do this? It's because proteins, they realize their function by interacting with one another, forming functional complexes. So basically, it's not like one protein has a function of its own, but rather it's the protein in combination with other proteins that realizes its function and now has a function. So unit of function is not really a protein, it's a complex. And for that reason, many of the drug targets are not just one protein. And if that protein is not doing a good job, we look for the protein that it interacts with. And it's through these interactions, it's that the protein realizes different functions. Just to give you an example of what I mean by that is that right now, I'm interacting with a bunch of scientists, which are you guys. That automatically turns me into a scientist. I'm talking about science. When I go home, I'm dealing with the kids, that forces me to be a father. So I have many functions, but these functions are realized by interacting with certain partners. So that's one reason why we study protein-protein interaction. And the next reason is that if we really want to understand the biology of a cell, for example, we want to understand the biology of a tumor cell, we need to study it in a systematic way. And protein-protein interaction is the major component of such systems. Okay, so at this point, I'm going to take a pause and I'm going to take many of us back a few years, maybe 1 or 2 years, in my case, maybe 5 or 6 years, back to when we started university. So assume that we are back at university and this is 3rd year of college and we go to the class, it's the beginning of the year. We kind of have an idea of who the students are because we have taken classes with them before. We have seen them in different labs, so we kind of know who are going to be our classmates. And all of a sudden, we see a new person, which is represented with a big, happy green face. And that person is sitting in one of our lecture rooms. And that person is interacting with a number of biochemistry students. He's also asking questions from the biochemistry TA. And the other day I was passing by the offices for the professors, and I noticed that this person is asking to get grades because he wanted to do something with the grades. And also I've noticed that he has interacted with the biochemistry administration staff. So that's all very good. I have not talked to this person. I don't know who this person is. I don't know his name. I don't know where he's from. I don't know how many languages he speaks. But just taking a step back and looking at all the interactions that this person is making, it's just very easy for us to conclude with certain degree of certainty that this person is a biochemistry student, just because of the interaction that that person is making. That's how we study protein-protein interaction. That's how we try to study novel gene functions. We want to understand what the function of a protein is. We look at its interacting partners. If the interacting partners are involved in process X, most likely this protein is also involved in process X. If I isolate this protein and look at its components, what interacts with it, I can say something about the function of this protein. Okay, so what are the tools that we use to study protein-protein interaction? The tools are nothing fancy, although the name protein-protein interaction has been very attractive in the past few years. It's nothing new. So I'm just going to go back. The techniques such as co-precipitation, immunoprecipitation, we have been using it for the past 50 years. Now, in the past 2 decades or so, we have had more advanced technology that we can apply them to genome-wide, and we can do high throughput investigations, such as yeast to hybrid and tap tagging. So what are those two? In a very simplified format, I'm explaining here what this yeast to hybrid is all about. So here we have a transcription factor, which is divided into 2 sections of X and Y. Now, if X and Y is dividing the transcription factor, transcription is not taking place. But if they're brought close to each other, then transcription is going to take place. Now, using yeast to hybrid, the question that I'm asking is that if the red protein, which is fused to X, can interact with yellow protein, which is fused to Y. And if there is an interaction, yellow is going to interact with red. It's going to bring Y closer to X. Transcription is complete. We're going to get a signal. In this case, the signal is beta-galactosidase. It could be any gene of interest, but a gene is turned on. So this is the basis of what yeast-2 hybrid is all about. There are many different versions of this. This is the most basic one. In the case of tap tagging, we get the protein of interest, which is shown in X. We double tag it, and using two-step purification to get rid of a lot of the contaminants, we isolate the protein of interest together with what's interacting with it. And the identity of the interactors are identified using mass spec. So back a few years ago, we identified 3 proteins that they were interacting with a number of transcription factors. And once we put together all of these interactions, it gives us this very messy diagram. Many of it is real, many of it is not. And the big challenge, I put this diagram together just to give you an idea of the challenge of protein-protein interaction, is to really clean up this data and see what is meaningful. And just to give you a little bit more of a hint, is that this is a subsection of a subsection of transcription factors, which are forming these interactions. Okay, so the techniques that we are using, just like any other techniques, they have inherent advantages and disadvantages. Main disadvantages is that you cannot apply them easily to all proteins. Some proteins do not like to be tagged. Their function changes when they're tagged. And there is high rate of false positive and false negative associated with all of these techniques. And of course, they're expensive. They take a lot of time, et cetera, et cetera. But the biggest problem is that you get a lot of false positive and a lot of false negatives. Now, when you compare the 4 gold standard genome-wide investigations using yeast-2 hybrid, 2 of them yeast-2 hybrid and 2 of them tap tagging, and you compare these interactions, they have identified thousands of interactions, but interestingly enough, only 24 of them are shared among all of them, just highlighting how much garbage we have in this data. The fact that we have a lot of garbage in this data provides opportunity for computational methods to come and clean up this data, or also try to predict what interactions might be all about. Now, computational biology, it's nothing fancy. Artificial intelligence, it's nothing fancy. Again, we are hearing on artificial intelligence in the news a lot these days, but it's really, if you ask me, it's nothing fancy, too fancy. Almost 80% of computational biology and artificial intelligence is looking for patterns. I can look for patterns myself with my eye, and I can get the computer to do that for me, as long as I know what pattern I'm looking for. I can train the computer to do this for me. From one set of data, it can learn to do it better in the next set. And then it's going to improve it in the next set. And that's what artificial intelligence is all about. I personally was never a computational biologist. I'm a geneticist by training. But what happened was that a good friend approached me and said, hey, let's do some work together. And that happened to be the chair of the computer science department at the institute when I joined. So we sat down and we communicated for 2 hours every week over 2 years. And at the end of it, what we realized was that the only reason that we couldn't work together was the language barrier. I thought I could understand what he was telling me. He thought he could understand what I was telling him. But next week we would get together and we couldn't understand each other's language. So after we realized that, hey, now we have a good feeling that we can understand each other's language, that's the time that very fruitful collaboration started. So there are a bunch of programs now that they can predict protein-protein interaction. For example, on the top left corner, the way that this pattern finding works is that it looks for 2 different proteins that during the course of evolution, they have moved together. And in some cases, maybe they have been fused. Therefore, based on that, it's going to predict that these 2 proteins are interacting with one another. If we come a little bit down in parts D, we have 2 different domains that we know they cause interaction. Fine. If we have similar domains to those in 2 independent proteins, we can predict that these similar domains are also causing an interaction. A very interesting one is C, which is what Rosetta is all about. For those of you guys who are more familiar with computational biology, it basically looks at the three-dimensional structure of the proteins. So if you have 4 fingers on one protein, and in the other protein you have anti-4 fingers, it just makes sense that these 4 fingers are made to click with the anti-4 and form an interaction. So basically that's the docking project, that if a protein looks like this, and another protein looks like this, that's the basis of an interaction. So the big question that at a time we wanted to ask ourselves was that, can we predict protein-protein interaction using primary sequence and primary sequence alone? Now, there's quite a bit of logic behind this, that why it should have worked, why it should work. I'm not going to get into that. But just to take you back a few years when we started this, that was the question for us. The primary sequence should have some information that could mediate interactions. And the good thing about this was that it was not based on any predefined information, such as the 3D structure of the protein. We did not, if this was the case, we don't need that. And we can apply it to all kinds of genomes. For example, COVID, the moment that we sequence it, we can look at all the interaction that it made. So it would have been beautiful if this works. So the working hypothesis was that there are interaction codes that, like a simple touch, that they come and they pass on a signal from one protein into another protein. And this is just like the example of Michelangelo's creation of Adam, that it's by magical touch, and the signal gets passed on to the other protein. And that's what we were working on. So this was the basis of what we wanted to tackle. And the big question was that we want to look for the patterns to see if there are any signals in proteins. Obviously, I could have done that myself. I could sit down and look at sequences and try to match them, but I have someone who has a lot of computation power. So we decided to ask the computer to do this basic work for us. So the basis of our original algorithm was extremely simple. We were looking for windows of 20 amino acids, and we were moving them 1 amino acid at a time. So for a given protein of, 200 amino acid length, we would have almost 200 minus 20 windows, and we would compare each of these windows to similar windows in the entire genome. Now, the fact that I say the entire genome makes you realize that there is a lot of computation in this. So it's not just we do simple matchup between 2 different windows. We would look for tiny windows of 20 amino acid in the entire genome. And the question that we were asking was that, are there any co-occurring pair that is found in the interaction set of proteins, which is absent from those proteins which are not interacting? So that's what the hypothesis was all about. And that's what this program was doing for us. And voila, it kind of worked, that we have 2 proteins in this case, that they have tiny regions. One of them is shown around 750 to 800, and the other one around 150 to 200. That seems to be co-occurring together a lot in the data set of proteins which are interacting with one another. This is in comparison to proteins that do not interact. They did not have these signals. So these signals, according to us, they probably were working as Velcros, that you can take a Velcro, add it to one protein, you can take anti-Velcro, add it to another protein, and now these 2 are interacting with one another. And we had the evidence for it. We were able to use this information and predict novel complexes that then we can confirm using tap tagging, affinity purification, and we could also break down the components of these complexes to see what are the major components and what's the topology of this complex. And the major components, when we delete them, basically we showed that the whole complex can fall apart. Those components that they are not playing a major role, if we delete them, the complex was still functional. So that's what we were doing at that time. Now, something very interesting happened was that the best confirmation that a scientist can get is that if other scientists take his or her approach and confirm it. So somebody decided, and that's Park's comparison experiment, a gentleman called Dr. Park decided to compare our algorithm to other competing algorithms out there. And in all the tests that he did, our algorithm was outperforming all other algorithms out there. So we were extremely happy about it. So that gave us a really boost of confidence that what we were doing was really meaningful and was getting us somewhere. But we had a big challenge because computationally, this was a massive work. For two proteins to analyze their interaction, it would have taken overnight. So if we wanted to do this in a simple model organism, such as Saccharomyces cerevisiae, that's the given model organism in systems biology, we have almost 20 million pairs. So that would result in 20 million days of computation with very good computers, which seemed impossible. So that was actually a very attractive computational problem for heavy-duty computational scientists. And they love problems like this because they're seemingly impossible to do. They try to do it in a very fast and efficient way. I'm not going to get into what they did, but they started parallelizing the program. We started using supercomputers. We used a lot of computational power, and we did a lot of pre-computational analysis. 2 PhD theses in computer science went to do this. And then after that, we made this happen, that for yeast, we can do the entire genome from 20 million days or so into 1 hour, which was fantastic for us. So now we had the power to do all-to-all computational analysis, which was great. Now, just to give you an idea of how massive this computation was that back a couple of years ago, we received an award from the government of Canada, from Compute Canada, which oversees all the computation projects in Canada. And the award was for the most amount of computation that was used for a single project, and that was given to us, which is not something to be very proud of because I don't know how many trees we burned. We had no idea we were burning the trees, but this was the most massive project that took place in Canada. Okay, so a little bit of computation here. If now we have massive amount of data, we need to kind of computationally look at it and see if it makes sense. This is called a ROC curve. And the way that it needs to work is that if we are doing almost perfect job, our curve should get very close to 1, which is on the top left corner. If we are doing rubbish work, it should get to 1 at the bottom right of the corner. And as you can see, just looking at the massive amount of data that we produced, they were pushing to the 1 on the top left corner, which was very good for us. And again, that was the booster that from the computational point of view that what we are doing made sense. So the way that this works is that if we predicted, let's say 20,000 interactions, you remove one of the interactions and you use the gold standard to try to predict that. And then you do the same thing for thousands of times and you come up with curves like this. So basically, we have a large data set of proteins interacting with one another, and we can publish it like this. But that's basically not very useful for biologists, for biochemists, for cell biologists. We need to do a little bit more. And we really need to show that there is at least another line of evidence that supports some of these interactions. Again, because we want to make sure that we are not publishing garbage. Now, when we looked at 14,000 of the novel interactions that we had, 8,500 of them, we were able to use at least another line of evidence to show that these two proteins should be interacting. What do I mean by that? So one of the evidence that we used was third-party interaction, which means that if A interacts with B and B interacts with C, we can kind of predict that very likely A might interact with C too. And we had that as part of the evidence for what we have produced. So that was very good for us. At the same time, if we are producing this data, we should be able to show the overlap that we have with the gold standard. And gold standard is tap tagging versus yeast-2 hybrid. And comparing the data that we generated, we had good overlap with both yeast-2 hybrid and tap tagging, as good as 2 different experiments using tap tagging with each other. So again, that was giving us more evidence that what we are publishing is pretty good, what we have identified. Now, looking at the distribution of interaction For example, in this case, we are looking at the membrane protein. In brown, this is the information that we have added to the knowledge, and the rest of the information which was added with other publications is shown at the bottom. So we were extremely happy about this, that we can actually produce a lot of data, and the trend of this data is identical to the experimental one. But we are doing it much faster, much cheaper, and we feel that it was way more efficient because we identified so many other interactions. Now, when we compared, this is the comparison that we did ourselves, we compared our technology to a few other technology out there that they were also predicting protein-protein interaction. And this was actually done because one of the reviewers in one of our papers asked, well, fine, this is, you guys are doing this fantastic, but how do you compete with others? So I want to bring your attention to very high specificity, which is at 99.95%. And no matter what we do, we are doing much better than the 3 leading computational programs that they were working based on other bases. So we were doing folds better than them in producing high quality protein-protein interaction, which was, again, very good. So having said all of that, we can put a nice pretty picture of interaction together, but what is it that we can use this interaction for? Like, what's the guts of this? This is all beautiful. We can understand how cell may respond to internal and external stimuli, but how do we use it? The way that we use it is that, generally speaking, we look for a new player in the same old pathway. So in this case, assume that all the proteins which are here and shown in this complex in red and blue, with the exception of this one, are involved in cell transformation in breast cancer. Now, when we are looking at protein-protein interaction data, we see that this new protein is also interacting with a bunch of this previously known complex. Just because of the interaction that it's making, we can predict that it's involved in the process of interest. And that's exactly what we started doing. So this is the underlying principle. Look at the interactions, and based on the interactions, come up with a function. So in the next few slides, I'm just going to go over some of the examples that we have used. And again, this is just for discovery based. We were interested in the process of double-stranded DNA break repair. This is the most severe form of damage at the DNA, that the whole DNA, both strands, falls apart. Now, the way that cell handles this is through homologous recombination that uses a template and brings the 2 pieces together, or non-homologous enjoining, that's just bring those 2 pieces which are broken apart and glues them back to each other. So we were interested in this process, and we said, okay, fine. What is it that we can do? The assay that we used is extremely simple because we are doing functional genomics. We are doing a number of them. We want to do many genes at the same time. So we need simple assays. So the assay that we used utilizes a plasmid that we can cut at a given restriction site. So we have the intact plasmid as well as the linearized plasmid. We transform them into the cell and we see how many colonies they form. If the cell can repair the broken DNA, we get the same number of colonies. But if the cell is not able to repair this DNA, we are going to get less colonies. And that's the way that easily we can select for those genes which are affecting the process of non-homologous end joining. So this is really the data that I wanted to show you. In this data, blue dots are those proteins that we know they are involved in the process of non-homologous end joining. Red is the interactions that we have identified. Yellow dots as well as the green dots are the new players that we see, that we claim they are involved in this process. And we apply them to the plasmid repair assay, and 50% of them show that yes, they reduce the efficiency of non-homologous end joining. So that tells us that using a very simple assay that might have a lot of limitation and cannot really detect a little bit of differences, we have at least 50% efficiency to detect proper function. But this is for DNA damage. Can we do it for any other processes? translation control is another process which is also involved in the development of certain cancers. So we figured, hey, can we detect new translation genes that are controlling translation? So from that, we identified 3 different genes. And when we applied them to different assays for translation fidelity and translation rate, we showed that yes, 100% of the genes that we predicted are involved in this process. So that was another boost for us. We can also detect markers for seasonal allergy for kids. So we hypothesized that there are 3 markers for seasonal allergy, and using microarray data, we confirmed that 2 of them seems to be real. Similarly, when we go to the breast cancer, we identified 3 new markers at a time, which is shown in kind of pinkish here, that they should be associated with this process. This is a very simplified diagram for breast cancer. And yes, now we know that these 3 markers are there. Good. So this concludes the first part of the talk. So I talked about what alternative protein-protein interaction is all about and how it got established. In the next part of the talk, I'll be talking about, now that we have this knowledge, can we apply it to something that we can use? Now, in the classical way of looking at proteins, proteins interact with one another using large 3-dimensional regions. So it's just like you and I, we can interact with one another using our arms. We can grab each other and give us a big hug. So this is the classical way of looking at protein-protein interaction. But now we know that proteins can also interact using smaller regions. So that would be just like a handshake or perhaps a kiss. And we all know that the effect of the kiss could be way more profound than a hug itself. So it's not like the hug is more meaningful than tiny pieces. These are just alternative. So now here's the important part. I need you to take a step back and look at these 2. And in the classical way that proteins are communicating with each other, the way that we can describe it is that they are speaking to each other in English language. Now, all of a sudden, we have discovered that there's another language that they use for communication with each other. And that language could be Spanish. And now here's the important part. So if by now I have bored you, the next 2 minutes is going to be important. So right now, we decided that in the classical way, proteins are communicating with each other using English language, but now proteins can also use Spanish to talk to each other. If we can understand the alphabet of Spanish, we should be able to reverse engineer this and generate proteins with a specific functions. So again, if the alphabet is completely understood to us, we can use that alphabet to reverse engineer proteins, or specifically peptides, that have a binding profile of interest. We can make molecules that specifically bind to protein of interest. So we can make specific drugs. What is a drug? In a very simplified manner, drug is a small compound that specifically interacts with an important site of a pharmaceutically important protein target, and in most cases, deactivate it. Sometimes it activates it. Now, this small compound, the problem is that not only it binds to this protein, it's going to bind to so many other proteins, and it's going to have side effects. So if somebody asks me, what's the difference between poison and drug? I'm going to come back and say nothing except the dose. If you use a little bit of it, that's a drug. But if you use a lot of it, that becomes poison because it has so many side effects. That's why nowadays, the drugs that are based on proteins and specifically antibodies are becoming very attractive because they seem to be more specific to that protein of interest, avoiding binding to the rest of the proteome. Now that we have this technology, if the technology is good, and if we fully understand the alphabet of Spanish, we should be able to make peptides which are working as good as antibodies. So that's something that we can use this technology for. And another thing is that, hey, antibodies are basically proteins which are designed to specifically bind to a molecule of interest, a pharmaceutically important molecule of interest in a soup of different proteins. We can make antibody-like molecules, and we can, in theory, we can make them way more efficiently because we can make small peptides. So again, if we understand this language and this alphabet, what is it that we can use it for? We can do therapeutic development, we can make drugs, we can do protein drug repurposing and looking at the side effects. So I'm not going to get into this, but if you're interested, you can ask me a question later or just email me. You can find my email on Google very easily. We can use these peptides for targeting. They are very small peptides. We can bind them to a toxin, and these peptides can deliver our toxin to the site of interest. And we can do anything that antibodies can do. Basically, we can do detection and diagnostic, as well as any other purpose that you can think of. Okay, so let's do the reverse engineering and let's see if something like that can work. Okay, in order to tell you what we have done and how our AI system works, I think the best is to talk a little bit about CellX. So in the case of CellX that many of you are familiar with, we are going to start with a random pool of nucleotides, DNA sequences, and we dump them into the system. We have a target in mind, and the pieces of DNA that bind to the target of interest are pulled out. Everything else is washed away. What we have pulled out is amplified, mutated, and then dumped back into the system. And the cycle continues for, I don't know, 10 to 20 rounds of purification and selection. So that's what CellX, and it's based on DNA. Now, the way that one of my students described the difference between CellX and what we are doing, he tried to describe it based on this diagram. I cannot do it as well as he does, but he was one of my favorite students, Dr. Dan Burnside. So I'm going to try to use his analogy for you. So this is a picture of Dan, and he has gone fishing with a bunch of friends, and he seems to be having, it seems like he's drinking beer more than he's doing fishing, but that's what he's doing. And eventually a fish is going to come into the picture. They are there just to have fun. Now, if you're doing this targeted, and you're doing this instead of 10 to 20 cycles, thousands of cycles, you are being a very professional with all the equipment. So all the cameras are there, the hook is there, and you're there to catch a fish. So that's what our program is all about. So the way that this algorithm works is that it starts with a bunch of random amino acid sequences, and it asks the question, do we have a sequence that may bind to the target of interest? And if it binds a little bit, it selects it out, mutates it, enters it into the next round, and try to make it better and better each round. Not only it's doing that, it's doing a negative selection too. So the program that we have, you can actually tell it that, hey, I want you to make a peptide that binds to this protein. At the same time, I want to make sure that this peptide does not bind to these proteins. So really, we are doing it very specifically, that this peptide is binding to the protein of interest, but nothing else. Good. So in order to do that, the first thing that we did was that, hey, let's take two targets and let's try to make peptides for it. These two targets should be an average everyday protein that it's out there that we can easily work with. And let's see if we can make these peptides. So that was the proof of concept for us. Of course, we went to yeast because it's much easier to do. We took two yeast proteins, PIN4 and PSK1. And we subjected these to our algorithm. And within 8 weeks, at that time, within 8 weeks, we produced peptides that they are supposed to bind to PIN4 and PSK1. Okay, so now that we have designed this peptide, we want to show that these peptides are actually binding to the target. So for that, we used an array in vitro array. So this is an in vitro technique that shows that two pieces of protein are interacting with one another. And we show that, yes, our peptide interacts with the protein of interest exactly at the site that we predicted. So that was beautiful for one of them. That was anti-pin4. And we measured the KD value to be in nanomolar. I think in this case was 7 nanomolar. For the next one, it was 10 nanomolar, the KD value. And again, it was interacting with the exact site that we were designing the peptide to bind to. So fantastic. But that was all in vitro. If it happens in vitro, it doesn't necessarily happen in vivo. So we wanted to do some in vivo investigation too. And we performed yeast-2 hybrid. We performed 2 different types of yeast-2 hybrid. And we showed that, yes, in vivo, our peptide is also interacting with the protein of interest. I'm not going to go over the data. Data is very clear. So just take my word for that. The next thing is that, hey, if you're using these peptides to bind to the protein of interest, can we do something to its expression? We made GFT tag for the target proteins, and we measured their expression. And we observed that when we induce the peptide of interest, the expression of the gene is significantly reduced in both cases. So again, that was very good. But really, the fantastic thing for us would be the deactivation of the target. And in this diagram, I'm going to use one of the examples to describe what's going on. So the first 2 lines which are shown up here, they are the controls. And the 2 down here, one of them, we have completely deleted the protein of interest. And in the other one, we have deactivated it using our peptide. And they're almost indistinguishable. So basically, that shows that the phenotype that we get is very much similar to the phenotype of a complete knockout. That's not even a knockdown. We deleted the entire gene. We saw a phenotype. We deactivated using our peptides, and we see the exact same phenotype in both cases. So that's fantastic. That was the proof of concept for us. So it happens that COVID kicks in back, I don't know, a year and a half ago. And just like everyone else that started working on this, we decided that, hey, let's use our technology to see if we can make anti-COVID peptides. And this is the diagram that you have seen so many times, and by now you're probably very tired of it. But very briefly, we have the spike protein on the surface of the SARS-CoV-2, and the spike protein interacts with ACE2, and that's how infection takes place. So if we look at the structure of the S protein, there are 2 regions which are important. One of them is the receptor binding domain, and then the other one is the S1, S2 cleavage site, which is a site of TRMPS, which cuts it into 2 pieces, and both of them are very important for the biology of the COVID. So what we did was that we said, okay, fine, we have this technology, we can make peptides against certain proteins. Not only that, in many cases we can do to exact regions of interest. So let's do that. And that's exactly what we did. So these were the 2 targets of interest. So that the way that we tackled this project was that we designed 22 peptides. Actually, we did a little bit more, but the time that I put the slide together, that's the number that we had. So we kind of kept it like that. Nothing has really changed significantly. 12 of them was for one side, 10 of them was for the second side. And we subjected these to 2 different assays for deactivation purpose, as well as for detection assays. So in the case of deactivation, we use Cynobiology kit, which was for SARS-CoV-2. So that's free advertisement for the sponsors. So we use their kit. And the way that this one of their kits works is that if the S protein interacts with ACE2, we get a signal. And if our peptide or a compound that you have inhibits this interaction, the signal is reduced. So the first thing that we observed was that for 2 of our best peptides, the signal basically was destroyed. And we were very surprised. We thought something has gone wrong because we couldn't get any signal. We reduced the volumes, this, that, and no, it was clear cut that this was basically killing the entire system, and no signal was given to us. So then that was in vitro assay. We also developed an in vivo assay. We wanted to know what's going on inside the cell. So for that, we divided a luciferase into two segments, the small bit and the large bit. And if the small bit would come close to the large bit, we get a signal. And that would be mediated using the RBD region and ACE2 because they have affinity for each other. That's part of the S protein. If they come together, this is very similar to yeast 2 hybrid. If they bring them close to each other, we're going to get a signal. Good. Now we introduced our peptides of interest, and we noticed that the signal was completely gone, was very much reduced, very much similar to the background. So again, now we had in vivo assay confirming that our peptide is deactivating the interaction between S protein and ACE2. But really, the ultimate proof was that if we go to the live virus, what's going to happen? So this data is with the live virus itself. It's not a pseudovirus. This is actually SARS-CoV-2. And we observed that it can reduce the replication of virus or infection by up to 80% for these peptides. So that was fantastic for us too. Now, for the detection part of it, we decided to use ELISA. Now we have indirect ELISA, for which you have your S protein, which is attached to the surface, and you need to have an antibody-like compound that can recognize this and give you a signal. In the case of sandwich ELISA, you need to capture your ligand of interest, which is again the S protein, and then detect it. So you need two antibody-like entities. So the first thing that we did was that we figured that, hey, we should be able to design antibodies that they have the same property. So why should we go to commercial antibodies? Our peptides should work as well as them. Let's do it. Let's find out. So we made a number of peptides, and we showed that 50% of these peptides were capable of detecting the S protein, which was fantastic for us because, again, that 50% was the key word for us, that we are at least 50% good in what we are doing. But really, the best experiment that made us very happy was that we have two sets of peptides. One of them binds to the RBD region, and the other set binds to the S1, S2 cleavage site. So we can capture and we can detect de novo. We don't need anything else. Can we do that? We used our peptides, and we managed to both capture and detect. So that was fantastic for us. Now, the numbers that I represent here was in buffer. If we go to cell lysate, we can go down to 100 picogram of the S protein in 1 ml of the cell lysate at standard concentration. In this case, 1 picogram is being detected, but really this is based on the background, which is water, the background of cell lysis. We can measure it down to 100 picogram per milliliter, and that's based on not optimizing ELISA. If we optimize the ELISA that we were doing, we should be able to detect it much, much better. But for our purposes, that was the proof of concept again, that we can target pharmaceutically important targets, and we can detect them. And in previous experiments, we can deactivate them. Good. So now we established that this technology is working very well, and these are the examples that we have. So what are the things that we can do in future? I have already talked about the peptide drug development, so we can use them for therapeutics. We can use these peptides for detection, diagnostics. Now, another interesting project that we can tackle is that we can go after pharmaceutically difficult and undruggable targets, such as RAS family of oncogenes. So RAS family of oncogenes, there were thoughts that if you manage to tackle them, you have a silver bullet for cancer. And the reason for that is that mutations in RAS family of oncogenes is found in certain type of cancers, 90% of them. And in up to 30% of all tumors have mutation in these RAS oncogenes. The problem is that the surface of these proteins are extremely smooth. So if you try to see if there is a compound that can bind to this, it just basically bounces back. So now what we can do is we can design certain peptides that could bind to RAS and in theory could deactivate it. And that's what we would like to do. We already have some indication that yes, our program can make peptides that binds to this. I have a new student who's going to work on this, but it's going to take some time to follow up with this. Okay, what else can we do? Targeting molecules for delivery of toxin. So antibodies, as we speak, are being used in many different companies to direct certain toxins to tumor cell. Now, in our case, we can make peptides, which can do the same thing. And our peptides are much smaller than the smallest of these antibodies. So again, that should have the advantage. That's something that we can tackle. We can also make bispecific or trispecific peptides. We have bispecific antibodies that one wing binds to the T cell and one wing of it binds to the tumor cell, bringing T cell close to the tumor cell. We can do exactly the same thing using the peptides. And again, the peptides are smaller and we can do this efficiently. But one project that I'm very excited about is that we can develop pairs of peptides, or basically we can make peptides against the entire human genome. So now we have the capability to tackle the entire proteome and say, hey, there's 22,000 of them out there. I'm going to make 2 pairs of peptides against all human proteomes. And at the end of this, this is going to give me a resource that is going to make it very unique and extremely powerful. Now, to tackle this, the first thing that we would like to do is to tackle the essential proteins at a very modest rate of 15 to 20 proteins per week. That's going to take us about 2.5 years, less than 3 years. So we are very excited about that, and we'll see how far we can get. But we don't have all the knowledge. We don't have the technology. So we are looking for people who would like to work with us. We are more than happy to work with them. We are in an academic setting, so we have access to so many different things, much cheaper than in industry. We are looking for the right partner to see what we can do. Of course, this is not my work. This is the work of fantastic group of students and collaborators. Specifically, I would like to mention Dan Burnside and Baron Salmanfar, 2 fantastic former students and current colleagues. I would like to also mention Frank Dene, who's a very good friend and collaborators, and we started this project together. He's the computer scientist that I was telling you about, as well as other collaborators that they have been fantastic, and the funding agency. And at the end, again, I would like to thank for the invitation and thank you all for listening to my talk. And at this point, I'll be more than happy to answer any questions.
Thank you, Dr. Golshani, for that excellent presentation. It's fantastic work that you've done. You're truly on the cutting edge of computational biology and predicting protein-protein interaction and optimizing peptide development. Just personally, I was delighted to see that you mentioned the yeast-2 hybrid system. I actually utilized that system back in graduate school in the early 90s and identified a transcription factor called paraxis, which is a basic helix-loop-helix protein interacting with E12 and E47, knocked it out of mice and realized it's a master regulator. And so I truly understand the value of yeast and yeast-2 hybrid.
Excellent.
So we got a ton of great questions here. I'm just going to jump right into them, starting with Edward. If I don't say your last name, it's because I wasn't able to see it on the screen. It's a very generalized question, and it is, how much do protein-protein interactions really contribute to drug discovery if the drug is a protein or a peptide? Could you just answer that generally?
If the drug is a protein, the next generation of drugs, they would be based on protein-protein interaction. The old generations, we were looking for compounds, and these compounds, they would bind to the protein and deactivate them. And we couldn't really tell what this deactivation is all about. But in the majority of them, the deactivation is that you're going to get a small molecule and it binds to the protein and prevents it from doing the function that it's supposed to do, and prevents it to communicate with other proteins. And it's basically as good as dead. So this is the old way of doing it. Now, the new way is that, hey, I know that this interaction is important for a certain case. So let me interfere with this interaction. That's actually a very good question. Traditionally, people would look for this target because everyone has talked about this target. Now, when the target gets saturated, they look for a second target, which is involved in the same pathway. Well, this is the second target interacting with it. So they start getting rid of that. If you get rid of the interaction between the two, this is the same thing. So if you ask me, this would be the future of drug development.
Right, great. Question from Rosanna. She wanted to know if you could cite some examples of well-known peptide drugs that have been approved by the FDA.
Not from the back of my mind, no. But there are many of them.
Right. Yeah, that's a tough one.
She got me there.
Yeah, she got me too. Ollie Fake, forgive me if I didn't pronounce your name correctly. He mentions what's been referred to previously as junk DNA. And specifically, we realize that there really is no such thing as junk DNA, and it's now actually referred to as the epigenome. Do you believe that there's junk protein or non-functional waste protein, or do you think every protein has a function?
Fantastic question. I also agree that there is no such thing as junk DNA. It does something. Maybe it came out just because I was doing it in a very simplistic manner. Now, junk DNA or random pieces of DNA or DNA which doesn't have massive function is the reason why non-homologous end joining is the dominant pathway versus homologous recombination in human cell. So it does play a role even if it were to be junk. Now, many of these are having regulatory factors. The question that you ask is that, are there any proteins that do not have, let me modify this, essential functions? And the answer that I'm going to say is that no. Every protein has some sort of an essential function, but we have not managed to find where that protein is needed. See, right now we come down and say, hey, we have 2,000 essential proteins. These 2,000 are essential proteins the way that we are using the cell. But if we use the cell under different conditions, we're going to find out that other proteins are important too. So I don't think there is any such protein as non-functional. So all proteins, they have a function because it just doesn't make sense. Why bother spending so much energy on it?
Right. Sounds great. I totally agree. Next question is from Shruti Uni. Can your pipe computational technology distinguish between non-specific and specific protein-protein interactions?
Yes, it does. So basically, we can use it as a filter to clean up protein-protein interactions that other people tackled. And also we can detect de novo protein-protein interaction. We can use it both ways.
Great, great. Xavier Arias asks, are non-conventional interactions just transient, or can they be more long-term and permanent and perhaps even covalent, for example?
Covalent, I would be surprised. There is no evidence for that, but there is no evidence to suggest that they're transient. They can stick together and they seem to be very strong in certain cases.
Great. And Xavier also asked, what's the average size of a peptide drug? Like how many amino acids?
Okay, we have peptide drugs that some people would use, and they say they have something between 3 to 10 amino acids. For us, when I talk about these peptide drugs, the size is between 25 and 75.
Okay, great. And you answered Brandon Veltry's question about covalency. Chao Chen asks, can computational methods predict the epitopes that antibodies would bind to?
Sorry, what binds to?
In general, that an antibody would bind to. Could computational methods predict the epitope that an antibody would bind to?
It might. We haven't looked into this, but that's an interesting question because the antibody is a big molecule, but only a small part of it is what binds to the target protein of interest. What we are proposing is that we can make that little piece. So that little piece is just like what we are making. So at some point, we haven't tackled that. We haven't looked at the antibodies and tried to predict what they bind to because that was not part of our interest, but we can do that. So if you get the sequence, the sequence of interest, we can apply it to the genome-wide and see where it binds.
Fantastic. Mark Whitmer asked, do your peptides ever include non-natural amino acids?
Good question. And the answer is no. We decided to stick with the natural ones because if you want to make them, it's much cheaper.
Got it. Great. Mohammed Ibrahim asked, do COVID spike mutations from new variants showed significant impact on spike epitopes S1, S2, results on vaccine escape, on dry lab data, or wet virus neutralization? I'm not sure I understand that question. Maybe you understand it, Ashkan?
I'm going to modify this to what I think he probably meant. He probably wanted to ask if our peptides can also work on the variants. Our fundings ran out, so we didn't really test this. And then the variants came into picture. But one of our collaborators says that he checked with one of the variants, and yes, it works on that variant too. And maybe while we are on this topic, I can also add the fact that we took one of the peptides that we were interested in that was working very